Hello everyone, now just quickly skim through the gene silencing and its application topic. Here we will be learning a quick recap about antisense RNA, RNAi, gene knockouts. I'm in a hurry, so I'm gonna quickly skim through this topic. Okay, what is gene silencing? Turning off a gene. This process happens naturally, occurs at a transcriptional or a post-transcriptional level. An important laboratory technique, epigenetic regulation of gene expression. Gene silencing is a technique that aims to reduce or eliminate the production of a protein from its corresponding gene. Gene silencing is a technique that aims to reduce or eliminate the production of protein from its corresponding gene. That is, the gene which would be expressed or turned on under normal circumstances is switched off the machinery of the cell. It occurs when RNA is unable to make a protein during translation. Gene silencing is same as gene knockdown, but is totally different from the gene knockout. When the genes are knocked down, their expression is reduced, whereas in contrast, when genes are knocked out, they are completely erased from organism's genome and thus have no expression. Processes of gene silencing. Genes are regulated at either transcriptional or post-transcriptional level. Transcriptional gene silencing is the result of histone modifications, creating an environment of heterochromatin around the gene that makes it inaccessible to transcriptional gene silencing is the result of histone modifications creating an environment of heterochromatin around the gene that makes it inaccessible to transcriptional machinery, RNA polymerase, transcription factors, etc. So epigenetic modifications will also be there. Post-transcriptional gene silencing is the result of mRNA of a particular gene being destroyed or blocked. So the destruction of the mRNA prevents translation to form an active gene product, in most cases a protein. The blocking of gene occurs through the restriction of mRNA prevents translation to form an active gene product, in most cases a protein. The blockage of the gene occurs through the activity of silencers which bind to repressor regions. A common mechanism of post-transcriptional gene silencing is RNAi. The discovery of the mechanism of RNA interference by dsDNA by Professor Andrew Fire and Professor Mello in 1998, Nobel Prize in 2006. How does it work? This is accomplished by binding a specific strand of RNA to an existing mRNA strand. An mRNA creates a copy of DNA strand. By binding the RNA to the mRNA is prevented from replicating that portion of the DNA. Specific genes can be targeted and prevented from replicating into new DNA strands. Methods of post-transcriptional gene silencing: antisense oligonucleotides, ribozymes, RNA interference RNA. Antisense RNA oligonucleotides. Antisense oligonucleotides were discovered in 1978 by Paul and Mary. So oligonucleotides, which are short nucleic acid fragments, bind to complementary target mRNA molecule when added to the cell. During dsRNA cleavage, different antisense RNA classes are produced: siRNA, miRNA. Antisense RNA case study: Flavor Saver tomato. The first FDA-approved genetically modified food licensed in 1994 will not soften while ripening on the vine. Increased shelf life, tomatoes can be shipped safely, keep their color, and have natural flavors. Before the flavor saver, picked before ripe and gassed with ethylene to give red color, keeps them from becoming spoiled. Tomatoes lose their taste and taste more like cardboard. First genetically engineered crop, flavor saver tomato, no alien gene, modified functioning of existing gene. Okay. Antisense RNA technology could not find acceptance due to poor genetic background. Okay. Improved shelf life. GM versus traditional tomato. The flavor saver tomato ripens on the vine, resulting in fuller flavor. It is modified so that it remains firm after harvesting. Flavor saver tomato, here we can see GM versus traditional tomato. The traditional tomato must be harvested while it is still green and then firm so that it is not crushed on the way to the supermarket. The traditional tomato is sprayed with ethylene after shipping to induce ripening, right? But decreased flavor. Okay, the flavor saver tomato ripens on the vine, resulting in fuller flavor. This is modified so that it remains firm after harvesting. Ripe and increased flavor. Flavor saver tomatoes, also known as CGN. This number, a genetically modified tomato, was the first commercially grown and genetically engineered food to be granted a license for human consumption. It was produced by the Californian company called G. Making of flavor saver: normal tomato, normal mRNA, sense PG, protein beats normal rooting tomato due to PG. Transgene tomato, PG gene, sense mRNA, antisense mRNA, complementary mRNA, antisense. Ripe tomato, no rooting for about 3 weeks. Enzyme polygalacturonase breaks down structural polysaccharide pectin in wall of a plant. This is part of the natural decay process in a plant. Flavor saver tomatoes have been constructed that express an antisense mRNA complementary to mRNA for an enzyme involved in polygalactose enzyme. These tomatoes make only 10% of normal amount of enzyme, thus delaying ripening production. So antisense RNA technology, flavor saver tomato, it blocks the activity of mRNA in a stoichiometric manner. So antisense RNA has the opposite sense to mRNA. The presence of complementary sense and antisense RNA in the same cell can lead to the formation of a stable duplex which interfere with gene expression at the levels of RNA processing. Possible translation: this technology widely used in plants for gene inhibition. Gene silencing method: antisense RNA oligonucleotides. Antisense oligonucleotides were considered in 1978 by Paul and Mary. Oligonucleotides, which are short nucleic acid fragments, bind to complementary target mRNA molecules when added to the cell. During dsRNA cleavage, different antisense RNA classes are produced like siRNA and miRNA. Okay. Then small interfering RNA, siRNA, short interfering RNA or silencing RNA, 21 to 23 nucleotide long double-stranded RNA can be exogenously artificially introduced into cells by transfection. Each strand of siRNA has 5' phosphate terminal, 3' hydroxyl terminal, 2/3 nucleotide overhangs. Micro RNA, semi-RNA, single-stranded RNA molecule, 22 to 25 nucleotide encoded by genes but are not translated into protein non-coding RNA. Primary transcript is processed into short structure called a pre-miRNA and finally into a functional miRNA. First miRNA is Lin4 microRNA, miRNA. miRNA, microRNA, miRNA originate from capped and polyadenylated full-length DNA precursors, RNA precursors, pre-miRNA. Hairpin precursors, mature miRNA. miRNA originates as ssRNA that forms a hairpin secondary structure. miRNA regulates post-transcriptional gene expression and is often not 100% complementary to the target, and also miRNA helps to regulate gene expression, particularly during induction of heterochromatin formation. Serves to down-regulate genes pre-transcriptionally. RNA-induced transcriptional silencing or RITS. miRNA-mediated gene silencing. Long double-stranded siRNA precursors, pre-siRNA. Short hairpin miRNA precursors, pre-miRNA. siRNA duplexes, miRNA duplexes. Perfect siRNA match with miRNA targets. Partial miRNA mismatch with the mRNA targets. The next, ribozymes. Ribozymes are catalytic RNA molecules used to inhibit gene expression. These molecules work by cleaving mRNA molecules, essentially silencing the genes that produced them. Sidney Altman and Thomas first discovered catalytic RNA molecule. Ribozyme, target RNA molecule. Ribozyme binds to and cleaves target RNA molecule at a specific site. Cleaved products formed by the cleavage of an RNA molecule with the ribozyme. Post-transcriptional gene silencing by double-stranded RNA, PTGS. Viral RNA, dicer, dsRNA cleavage, siRNA, RISC, virus-induced gene silencing, HpRNA, hairpin RNA, RISC DNA-induced silencing complex, then antisense strand, degraded mRNA. Overview of RNA-induced transcriptional and translational silencing. gsRNA, viral replication, precursor miRNA, siRNA, effector complex, RITS, RISC. RITS, genome RNA, DNA histone methylation, mRNA degradation, translation block. Types of gene silencing: transcriptional, genomic imprinting, paramutation, transposon silencing, transgene silencing, position effect, RNA directed DNA methylation. Once again, transcriptional, genomic imprinting, paramutation, transposon silencing, transgene silencing, position effect, RNA directed DNA methylation. Then post transcriptional, RNA interference, nonsense mediated decay, meiotic transfection, meiotic silencing of unpaired DNA. And next, transcriptional gene silencing in detail. It is the result of histone modifications creating an environment of heterochromatin around a gene that makes it inaccessible to transcriptional machinery, RNA polymerase, transcription factors, etc. Genomic imprinting. Genomic imprinting is a genetic phenomena by which certain genes are expressed in a parent to origin specific manner. It is an inheritance process dependent on the classical Mendelian inheritance. Genomic imprinting have been demonstrated in insects, mammals, and flowering plants. Genomic imprinting, here is an example of different genes where the maternal allele is always imprinted. Allele from the mother is inactive, father. Allele from the mother is inactive, mother, offspring. Maternal imprinting. Phenomenon by which certain genes are expressed in a parent to origin specific manner. If the allele inherited from the father is imprinted, it is thereby silenced and only the allele from the mother is expressed. Paramutation. Paramutation is an interaction between two alleles of single locus resulting in a heritable change of one allele that is induced by the other allele. Paramutation was first observed in the effect it had in the color of corn kernels in maize plants. The change may be in the pattern of RNA methylation or histone modification. Okay, the allele producing a change is said to be paramutagenic, while the allele that is been epigenetically altered is termed paramutable. Then position effect. Position effect is an effect on the expression of a gene when its location in a chromosome is changed, often by translocation. Position effect is an effect in the expression of a gene when its location in a chromosome is changed, often by translocation. Drosophila position effect, variegation with respect to eye color. These phenotypes are often due to the chromosomal translocation such that the color gene is now close to a region of heterochromatin. The heterochromatin can spread stochastically, randomly, and switch off the color gene, resulting in a white-eyed sect. RNA directed DNA methylation. RNA directed DNA methylation is an epigenetic process first elicited in plants whereby small double-stranded RNAs are processed to guide methylation to complementary DNA lozenge. The model plant organism is transposon silencing. DNA transposon end sequence, gene for transposon enzyme, another gene transposon enzyme. Transposon is cut out and inserted at new location. Disrupted gene. Transgene silencing. Unfortunate insertion of transgene into a transcription is an active part of genome. When an insertion of any transgene, it does not show activity as per desire, and it's because of its instability. The loss of transgene stability is because of gene silencing. Slow fruit softening tomato by reducing expression of polygalactoronase enzyme. Post-transcriptional gene silencing. The ability of exogenous or sometimes endogenous RNA to suppress the expression of the gene which correspond to an mRNA sequence. RNAi. RNA is a biological process in which RNA molecule inhibit gene expression or translation by neutralizing targeted mRNA molecule. It is post-transcriptional process triggered by the introduction of double-stranded RNA, dsRNA, which leads to the gene silencing in a sequence-specific manner. First evidence came from the studies on nematodes and Rhabdactis elegans and in Drosophila. Mechanism of RNAi initiation step double stranded RNA dsRNA molecule is cleaved to form 21 to 23 base pair double stranded fragments called short interfering RNAs, siRNA, passenger and guide RNAs.